Hello everyone. In this video we're going to make an experiment to extract DNA from cells. This experiment is required from you. You have to do it at home and you have to show me your experiment. You have to send me your video. In the video that you have to send, you have to explain step by step what are you doing. Also you have to send me a lab report. The main things from where we want to extract the DNA, we want to extract it from strawberry, kiwi, also you can use banana, but the most from other fruits, but from banana, from strawberry or kiwi, it's gonna be easier to see the strands of DNA. We have studied the structure of the DNA. How do we study that? What are the nucleotides? And I mentioned before, like the cell, it has like 2 meter long of the DNA. For now I'm going to show you 2 videos. These 2 videos will help you to do this experiment. Both of them, they are talking about extracting the DNA, but both of them help you to do this experiment. Let's start with the first one.
Okay, and then I'm going to put some salt in it. So what the salt's going to do is it's going to help break up the proteins that are holding some of the DNA together.
Here I'm going to stop. Here you have to use like salty water and start to smash the strawberry or the kiwi in salty water. Okay, look at this one.
So basically it's just going to help release the DNA easier. Okay, so now even though this looks mashed up, all of the cells that are in there are still whole cells. To break these up now, you can't do it physically, you need to do it chemically. And then you pour some of the soap in with the smashed
You add soap detergent to it. Look.
Strawberries and just mix it around for a while. Okay, now I just need to filter out the strawberries.
You have to filter it then.
Okay, and then you just pour your alcohol down the side.
Then you add to the filtered smashed or crushed strawberry or whatever kiwi, the thing that you want to use, you want to add alcohol.
Side of it. And you can see that stringy stuff forming in there. That's the DNA.
Can you see these strings, white things? You can use a toothpick to remove these
Here
things.
it
This
is.
is the DNA.
Strawberry DNA. So I used about the same mass of kiwi. It wasn't exactly the same. Let's pour in our alcohol. Okay, so this is my only distinct strand that I can get. Probably there wasn't enough to coagulate together to form a big chunk. Okay, let's see if we can get any DNA out of our shrimp now. Wow, look at that. So DNA is the instructions for the cells in your body. The DNA is how cells know what proteins to form and what to do with them. So DNA has a lot of information on that, and that's the reason that DNA chains are so long. In each cell in your body, you have around 3 meters of coiled up DNA. This is a ton. This is way more than the strawberry. I'm really surprised at this. Holy cow. Okay, and after thinking about this some more, the reason I got so much DNA from the shrimp, I should have known this from the start, but the shrimp has a lot of protein in it. And so the proteins also coagulate with the isopropyl alcohol. So really what I have here is a mixture of DNA and protein, but I'll still take it. So the DNA here doesn't look very long, and that's because the isopropyl alcohol made all the DNA kind of coil together and clump up into big, large groups. And so you don't have these long strands of DNA anymore. But DNA is actually extremely long. So you basically have these tiny little strings around 3 meters long in every single one of your cells. And that's why when you feel this, it feels kind of rope-like, stretchy. Okay, strawberry DNA. So no real taste to it. I could only taste a little bit of the rubbing alcohol, but other than that, no taste to it. The texture of it was just slimy. Okay, now this is the kiwi DNA. So this is only a tiny little amount. So I guarantee you can't see it. I can see it though, so you just have to trust me, it's on there. That one actually tasted good, kind of a concentrated kiwi taste. Okay, now my shrimp DNA. This is actually shrimp DNA mixed with protein, but actually not bad. That's actually pretty good. The texture of it was just kind of gummy, but it had a shrimp flavor to it. So that was pretty good. So is it dangerous to eat pure DNA? No, it's not. I'm actually worse off eating the isopropyl alcohol and the soap that was in it. And you eat DNA all the time whenever you eat anything. It's just not concentrated like that. Thanks for watching, and I'll see you next time.
Okay, now I'm going to show you another experiment that help you to know how to extract the DNA. The same thing, they are going to use here a strawberry. Listen carefully to this.
Did you know that you share 50% of your DNA with a banana? You might be more closely related than you thought. This is because all living cells contain genetic material in the form of the chemical DNA. Each of your cells contains an incredible 2 meters of DNA. It is possible to fit all that DNA in a tiny cell because the DNA is extremely narrow, only about 2 nanometers, and it is tightly wound around proteins. You can find out more about DNA and its structure by watching this video. Do you want to see what DNA looks like? Well, it is possible. You just need to extract it from some cells. You can do this in the lab or even in your own kitchen. The easiest way to do this is to use some fruit. In this video we will go through the steps to show you how. First, you need to choose your fruit. The best fruits to use are those that have plenty of DNA. Strawberries are a good choice, as are kiwi fruit. You then blend or mash the fruit with salt water. This breaks apart the cells from each other. It also starts to break down the cell walls. Salt is used so that the DNA is more likely to clump together in the final stage.
Okay, you have to know why we added salty water, why we add soap, why we add alcohol. All of these you have to mention why you add each one of these substances, okay?
The next stage is to pass the liquid through a sieve and collect the fruit soup. This will contain all the strawberry cells with the DNA inside the nucleus. To break apart the membrane surrounding the cell and the nucleus and release the DNA, you add some detergent, such as washing up liquid to the soup.
Soap, like this
This
of the other
ruptures the
video.
cell membranes. Adding a pinch of protease enzyme will help break down the proteins the DNA is.
Here, the protease enzyme, you can add, for example, apple juice, or it has inside it protease enzyme, but it's not very important to add it, okay?
Wound tightly around. The DNA will uncoil and will now be present in the mixture as long strings. You can use pineapple juice, contact lens cleaner, or a pinch of meat tenderizer. These all contain protease enzymes.
Okay, you know how from where you got the protease enzyme? From any of these, but it doesn't matter if you don't use it.
You can't yet see the DNA because it is dissolved in the mixture. To see it, you have to make it insoluble. To do this, you slowly pour ice-cold ethanol into the mixture. The ethanol is at this low temperature to slow down the action of enzymes naturally present in the cell that will break down the DNA. In a normal, healthy cell, these enzymes are usually kept separate from the DNA by the nucleus membrane, but you ruptured this in an earlier step. Ethanol is less dense than water, so it forms a layer on top of the fruit soup. The salted DNA is insoluble in ethanol, so it will begin to precipitate out of the fruit soup at the boundary between the soup and the ethanol. It transforms from a dissolved solute into an insoluble substance in the ethanol layer. You will see cloudy strings of DNA, which you can remove by winding it around a wooden stick.
Or toothpick you can
have
use.
extracted DNA. You can then observe this DNA with a microscope or use it for other experiments.
If you liked the video, give it a thumbs up.
So this is how to extract the DNA. I hope to hear from you and to see your videos how you are doing this experiment. It's a very fun experiment. I hope you find it fun also to see the DNA strands. Thank you for listening.